everyone. Have you ever wondered how a cell maintains its shape, moves, and transports materials inside it just like a well organized city? The answer lies in an invisible but powerful framework called the cytoskeleton. Welcome back to the channel BioBoost. We are starting a very important topic today from cell biology, that is the cytoskeleton. So, what is cytoskeleton exactly? It is a network of protein filaments present in the cytoplasm of all eukaryotic cells and even in a simpler form in prokaryotes. It is basically called as a skeleton and muscle of the cell, right? So it is a complex network of filaments and microtubules which forms a structural framework. Cytoskeleton is also responsible for segregation of chromosomes into two daughter cells at cell division. In this diagram here you can see how in a cell cytoskeleton is spread all over the cytoplasm. It is responsible for segregation of chromosomes. When we will cover cell division topic, there you can see the role of cytoskeleton. So when we talk about cytoskeleton, basically it is divided into parts where it is built on a framework of three types of protein filaments. In next topic we will cover these filaments. Here the first one is intermediate filament that you can see. Second is microtubule and microfilaments, which includes actin filaments. Their sizes are given and their shapes are also shown in these diagrams. Here in this cytoskeleton diagram you can see near the nucleus part how MTOC microtubule organizing center is present. So intermediate microtubules and actin, where microtubules ranges from 25 nanometer diameter, actin are of 7 nanometer diameter, and intermediate filaments are of 10 nanometer diameter almost. First we will see intermediate filaments. So they have great tensile strength. Their diameter is about 10 nanometer and they are made up of fibrous intermediate proteins. Means there are two types of structural proteins, one is fibrous and one is globular. So these are of fibrous intermediate proteins and their size ranges from 8 to 12 nanometer. In this diagram a type of intermediate filament is shown which is keratins. So keratins are coiled together in this diagram, which is also a type of intermediate filament. So they are made up of fibrous intermediate proteins. And here in this diagram you can see they are composed of two anti-parallel dimers. So they are basically dimeric. Dimers are present. Composed of two anti-parallel dimer. Now when we study about types, then they are basically divided into four types of filaments. Keratin filament. Next is vimentin filaments. Third are neurofilaments. And the last one, nuclear lamins. Now, when there is some fault or some damage in these filaments, then some disorders are produced in our body, in cells. These disorders of intermediate filaments are, first is epidermolysis bullosa simplex. So these disorders are mainly concerned with cytoskeleton damage or rupture or any kind of disturbance in their alignment. So first is epidermolysis bullosa simplex. Second is amyotrophic lateral sclerosis, ALS. These types and these disorders are very important from exam point of view, because these are asked in multiple choice questions where you are supposed to choose 2 to 3 disorders which are either are asked for cytoskeleton or any other cell organ related. So these you need to remember. Third was progeria. Next we will move to microtubules, which is second type of cytoskeleton. By the name itself you can see these are concerned with tubules, and with tubules a term is come forward which is tubulin. So these are long hollow cylinders made up of units which is known as tubulin. Single unit of these microtubules are considered tubulin. Their length is 25 nanometer in diameter. Their role in cytoskeleton is to provide internal shape and support to cell. So they basically provide internal shape and support to the cell. They are also involved in nucleic and cell division. So when a cell is under division, they are involved in DNA and cell division. And types, if we consider, then there are basically 2 types of tubulin. One is alpha tubulin and second one is beta tubulin. When tubulin heterodimers are formed, and when multiple tubulins are formed, then they form a protofilament, and these microtubules has plus and minus ends. Next cytoskeleton is cilia and flagella, which is also type of microtubule, just like tubulin. So cilia and flagella are basically hair-like structures. They are made up of microtubules and covered by extension of plasma membrane. So they are basically just an extension of plasma membrane. They are just extended form of plasma membrane. Cilia and flagella both move liquid past the surface of the cell. So their role is to move liquid whenever it passes or crosses the surface of the cell. So basically they are concerned with movement because they are hair-like structures. So they create current for obtaining food from aquatic medium, because in water bodies movement is needed. So these provide movement to the cell or to the organism. So on epithelial cell lining of respiratory tract, they have huge number of cilia, because we all know in respiratory tract air needs to pass through, so make a passage for the air. The arrangement here in this diagram you can see is of 9 plus 2, 2 in the middle, 9 pairs are around it. So around the central sheath you can see 9 plus 2 arrangement it is called. If there is an affected microtubule, then they get damaged. So these are the drugs that majorly affect the microtubules. These are Taxol, colchicine and vinblastine. These are also very important from exam point of view. Next, we will study the microfilaments. Microfilaments basically deal with actin filaments, which are the thin filaments of microfilaments. There are thin and thick filaments, out of which actin are the thin filaments. These are present throughout the cytoplasm of eukaryotic cell. These are present just beneath the plasma membrane, therefore they support the outer layer of cell. These are responsible for movement, especially involving cell surface, and they can form structures like microvilli on the brush bordered cells lining the intestine. Here you can see that actin filaments grow by addition of actin monomers, but in order to stop more addition of actin monomer, proteins such as thymosin and profilin binds to the monomers. When monomers are sequestered, then they are present there, and it is a reversible reaction where monomers get aligned together and they together form long filaments, actin filaments, and when their ends are needed to be blocked, which is also known as caping, then proteins such as thymosin and profilin are needed, which bind to these monomers, which block their further addition, further lengthening. So these ends are getting blocked here, and it is called as caping or end blocking. Next we will see how movement by actin filament is done. Because these filaments are required for movement, so here the cell pushes out protrusions at its leading edge. Means at the edge protrusions are present, which helps the cell to push out. Now these protrusions adhere to the surface and rest of the cell drags itself forward on these anchorage points. So basically a protrusion of the leading edge is made of actin filaments, then adhesion at the leading edge is done, and the body itself drags and they align on the anchorage points. Itself on these anchorage points. Next, we will move to another important topic, which is muscle contraction. We all know that each muscle fiber is a single long cylindrical muscle cell. Means a muscle fiber is a long single cylindrical muscle cell. Thick filaments of myofibrils are made up of protein, which is called as myosin, and thin filaments of myofibrils are made up of protein, which is known as actin. Now these organization of these filaments produce striations. Now here make a passage for the air, and this unit is known as sarcomere. So a sarcomere extends from Z line to Z line. Between two Z line, a single sarcomere is made. So it is just a rough diagrammatic representation of how a sarcomere is formed, which is a single unit of muscle contraction and relaxation. So basically this sarcomere is seen only when the muscle is in contracted state. these are the I bands, which are basically light bands, and they are made up of actin, and they are anchored to Z lines. A band is present, which is basically the dark band, and they are made up of overlapping thick and thin filaments, means actin and myosin filaments. In the center of A band is an H zone, which consists of myosin filaments only. This H zone is there, which is consisting of only the myosin filaments, and now sarcomere is done. Now we come to know about a theory which was given for sliding filament theory, which explains how muscle contract and relax at the molecular level. So this theory was proposed by Huxley and Hansen in the year 1954. The basic idea behind this theory was that according to it, muscle contraction occurs when thin filaments slide over thick filaments, which causes the muscle fiber to shorten without any change in filament length. And the structures involved are myofibrils, which are made up of repeating units called sarcomeres. We all know that each sarcomere contains actin myosin. Now we will understand the mechanism of muscle contraction. First, the beginning is from nerve impulse, where a stimulus is given which reaches the muscle fiber. Calcium ions are released from sarcoplasmic reticulum. In the last video, we know that sarcoplasmic reticulum stores calcium ions. So these calcium ions are released from sarcoplasmic reticulum. Now in this diagram here you can see calcium binds to troponin, which causes tropomyosin to move away. This exposes the myosin binding sites on actin. So myosin binding sites are open on actin. Myosin head attached to actin, forming the cross bridges. Now with the help of ATP, myosin heads pull actin filaments inward. Right? As actin slides over myosin, the sarcomere shortens. And this whole phenomena result in muscle contraction. Now things we need to notice is that the changes which is brought during this contraction include I band, which gets shortened, H zone, which disappears, A band, which remains constant, and sarcomere length decreases. You need to remember that filaments do not shorten, they slide. And in this process, ATP is very crucial. Calcium plays a regulatory role and A band length remains unchanged. So this was the whole theory of sliding filament. It is explained in a very simple and clear way. And if you find this video helpful, don't forget to like, share and subscribe to the channel. Thank you all for watching, and